Inside every single one of us, there's this ancient, vigilant defense force. It's a microscopic army that's constantly patrolling our body, deciding what belongs and what needs to be eliminated. Today, we're going to explore a quiet revolution in how we understand this force, and a discovery that might just teach us how to awaken its most powerful soldiers when they've fallen dormant.
And that's a really deep, and honestly, a kind of troubling question, right? These cells are supposed to be our very first line of defense against threats like cancer. So what makes them become unresponsive? And maybe more importantly, can they ever be woken up again? This is the mystery we're going to unpack together. Okay, so to really get into this, we first have to meet the cells that are at the very heart of this story. Think of them as the sentinels of our innate immune system, the ones who are always on guard.
So, they're called natural killer cells, or NK cells for short. Now, what's so special about them is that unlike a lot of other immune cells, they don't need to be trained to spot a specific enemy. They're basically born ready, with this innate ability to find and destroy stressed-out cells, whether they're infected by a virus or have become cancerous. They are absolutely crucial.
The thing is, though, sometimes these powerful sentinels, they're just not functional. For a long time, scientists have seen two different ways this can happen. Some NK cells are what's called anergic, which basically means they're naturally unresponsive, almost like they were born asleep and never woke up. Others become exhausted. They start out working just fine, but after long, drawn-out battles, like fighting a chronic tumor, they just get worn out and stop.
So, for the longest time, everyone just kind of assumed these two states, born asleep versus worn out, were totally separate issues with different causes. But some researchers started to wonder, what if that wasn't the whole story? What if, at a really deep molecular level, these two types of dysfunction were actually connected? What if they shared some kind of common identity?
And that one question, well, it kicked off a really deep investigation. Scientists set out to compare these dysfunctional cells, hunting for clues hidden in their genetic programming.
All right, let's take a look at the first big clue they found. What you're seeing here is a way to visualize the genetic activity of these cells. The healthy, working cells are all clustered over on the right, and the sleep cells are way over on the left. See that huge gap between them? That really shows just how different their whole genetic program is. At first, this seemed to confirm that, yep, these two cell types were just worlds apart.
But then came the moment that changed everything. The researchers took the genetic signature from their asleep cells and compared it to data from other studies that were looking at exhausted cells from chronic infections and cancer. And what they found was, well, it was astonishing. Even though they had completely different origins, the two types of dysfunctional cells looked incredibly alike. It turns out they found this shared molecular signature, a kind of ID. 3 genes in particular were basically working overtime in both the asleep cells and the exhausted ones. These genes, EGR2, DGK-alpha, and PDCD1, became the prime suspects. And that last one, PDCD1, you might have heard of it as PD1. It's a huge target in modern cancer therapy for releasing the brakes on the immune system, so finding it here was a massive clue that they were on the right track. So the easiest way to think about EGR2 and DGK-alpha is like their molecular brakes. When these genes are switched on, they act as an internal off switch. They actively hold the cell back, stopping it from launching an attack. It was as if both the asleep and the exhausted cells had their foot slammed firmly on the brake. So this discovery led to a really powerful what-if moment. What if you could just release those molecular brakes? Could you actually wake these cells up? It was time to put that idea to the test. And the experiment they designed to test this was, honestly, just beautiful in its simplicity. First, they took the asleep, anergetic NK cells. Then they used this technique called siRNA, you can think of it like a molecular mute button, to specifically silence that EGR2 brake gene. It's like cutting the power to that internal brake. And finally, the big test. Would these reawakened cells now do their job and kill tumor cells? Okay, so let's look at what they found, starting with the before picture. This chart is basically a measure of how good the cells are at killing tumors. The higher the bar, the more effective they are. And if you look at the far left, those are the untreated anergic cells. Their activity? It's pretty much zero. Just as expected, they were completely dormant.
But now, look what happens after they silence that EGR2 gene. That middle bar tells the whole story. The killing activity just shoots up dramatically. It's incredible. The once sleeping sentinels are now wide awake and just as lethal as healthy active cells. This was it. This was the proof that this dormant state wasn't permanent. It could actually be reversed.
I mean, this was such a remarkable finding. It didn't just solve a biological puzzle. It basically opened the door to a whole new way of thinking about therapy against cancer.
Okay, so waking up cells in a petri dish is one thing, and it's a huge step. But the real test, the real challenge, is can you do this inside the body, inside the incredibly complex and hostile environment of an actual tumor? Could the same idea work to re-energize the exhausted cells that are already there, fighting a losing battle?
So to figure that out, they set up this really clever experiment using a mouse model. First, they implant a human tumor. Then, a bit later, they inject human NK cells, which, just as they predicted, get exhausted by that tumor environment. And then, using these tiny little nanoparticles, they deliver the EGAR2 silencing therapy right to the tumor. Finally, they just watch and measure what happens.
And the results? Well, they were just striking. This graph here is tracking tumor growth over time. That top line, that's the tumors growing pretty much unchecked in the control group. But that bottom line, that's a completely different story. In the mice that got the therapy, those reawakened NK cells got to work, and you could see tumor growth slowed way down. They were fighting back from the inside. And this chart really puts a number on that effect. Just look at the difference. The tumor growth rate in the mice that got the EGFR2 reprogramming, it was cut by more than half compared to the controls. That's a huge, clear signal that releasing that one single molecular break makes a massive difference.
So here's the real takeaway, the crucial point from all this. Just like the researchers said, being asleep or exhausted isn't some permanent dead end for these cells. It's actually a programmable state. And that totally reframes how we think about immune failure in cancer. Not as a final defeat, but as a condition that just might be reversible. And that really leaves us with this incredibly hopeful and profound question. This research it's like a blueprint for a whole new kind of immunotherapy. Not just adding more soldiers to the fight, but reaching inside our own immune defenders and just flipping a switch, waking them up so our bodies can do what they're designed to do, heal themselves.